So students, in the previous session, I had explained you about the formation of the DNA or RNA for that matter, the nucleic acid, what all it consists of and how is it formed is what we have learnt in the previous session. So you need to remember what is nucleoside and how it is formed, right? Nucleoside means the pentose sugar. It can be either the ribose sugar in the case of RNA or it can be deoxyribose sugar in the case of the DNA. Right? So if this pentose sugar is attached with a nitrogen base to the first carbon atom with the help of the bond called N-glycosidic bond, then it will be called as nucleoside. So to this nucleoside, when a phosphate group is attached to the fifth carbon atom with which bond called as phosphoester bond, then it will be called as a nucleotide. Right? So when a nucleotide is formed, many nucleotides, they come together and form a chain of nucleotides. It's also called as a polynucleotide chain. During the formation of the polynucleotide chain, you know that the phosphate group present on the fifth carbon atom of one nucleotide will form another phosphoester bond with the third carbon atom of the next nucleotide and thus forming a phosphodiester bond. Okay, so ishtu matrana ನೀವು correct ಆಗಿ clear ಆಗಿ ನೆನಪಿಟ್ಕೊಳ್ಳಬೇಕು. This is the entire structure formation of what the DNA or RNA. Right? Now in today's session, let us study more about the DNA, and the DNA is well studied, it is well understood by the model called as double helix model or a double helix structure. See, it's a very famous, very well-known model even till today, and it was proposed by two well-known scientists, and they are named as they are called as James Watson and Francis Crick. See, these two people, they proposed, they found out, they discovered a new model. They introduced a new model that was called as the double helix structure or double helical model of the DNA, with which they could explain the entire structure in detail of the DNA. Right? So, how the discovery was done? That means before finding out this one, you already have learnt in the principles of inheritance chapter about various concepts of genetics, about the gene, all that you have already learnt. But how could they arrive at this particular conclusion? That means before this, there must have been so much trial and error things. They must have tried a lot of things. They must have experimented on a lot of things. See, Friedrich Mischer, a scientist by name Friedrich Mischer, in the year 1869, he was doing some experiment with the cells of the pus. You know the pus cells, WBC. Whenever a wound is there on your skin, so if the wound is still there for a very long duration, then pus creation will happen, right? So from the pus, what cells will be coming out is WBC cells, white blood corpuscles. And you have very well studied and understood in the first PU itself, WBC cells have got a larger nucleus. Right? So WBC, he accidentally isolated the nucleus, and from within the nucleus he isolated a material, white kind of material from the nucleus. Nucleus ಒಳಗಡೆಯಿಂದ ಒಂದು material ಅನ್ನು isolate ಮಾಡಿದ್ರು, and that material was called as nuclein. He named that material which was isolated from within the nucleus called as nuclein. Nucleus ಒಳಗಡೆ ಇತ್ತು, it was inside the nucleus, and that is why it was called as nuclein, right, or nuclein. Later, biochemical studies of this material proved that the acidic nature, so later by the biochemical tests, it was confirmed that it is acidic in nature, and that is why it was later called as nucleic acid. Right? So nucleic acid, further study, further biochemical analysis also gave us the information that there are two different types of nucleic acids. One is called as the DNA and the other is called as RNA, and this is how the discovery of the nucleic acids took place. And after that, see, James Watson and Francis Crick in the year 1953, They gave the double helix structure and explained entire thing about the double helical structure. But their model was based on, there were two main bases for their model. ಯಾವ ಬೇಸಿಸ್ ಮೇಲೆ ಅವ್ರ ಮಾಡೆಲ್ ಪ್ರಪೋಸ್ ಮಾಡಿದ್ರು ಅಂತ ಹೇಳಿದ್ರೆ, ದ ಫಸ್ಟ್ ಬೇಸಿಸ್ ಈಸ್ ದ ಎಕ್ಸ್-ರೇ ಕ್ರಿಸ್ಟಲೋಗ್ರಫಿ. X-ray crystallography of the molecules. See, you know that X-ray, when X-rays are passed through the body, what will be seen? The picture of the bones will be captured, isn't it? See, to study the molecular structures, the X-rays also are used. The molecular structures can be clearly seen, clearly studied, and clearly captured with the help of the X-rays. And that is what was done by two different scientists, Maurice Wilkins and Rosalind Franklin. ಈ ಇಬ್ಬರು ಸೈಂಟಿಸ್ಟ್ಗಳು, ಮಾರಿಸ್ ವಿಲ್ಕಿನ್ಸ್ ಅಂಡ್ ರೋಸಲಿಂಡ್ ಫ್ರಾಂಕ್ಲಿನ್, ದೇ ಹ್ಯಾಡ್ ಕ್ಯಾರಿಡ್ ಔಟ್ ದ ಎಕ್ಸ್-ರೇ ಕ್ರಿಸ್ಟಲೋಗ್ರಫಿ, ಅಂಡ್ ದ ಸ್ಟ್ರಕ್ಚರ್ ದಟ್ ದೇ ಹ್ಯಾಡ್ ಗಾಟ್ ಔಟ್ ಆಫ್ ದ ಎಕ್ಸ್-ರೇ ಕ್ರಿಸ್ಟಲೋಗ್ರಫಿ paved the way to propose the double helix structure by James Watson and Francis Crick. Right? And these people, they also shared a Nobel Prize for their discovery, for their proposal of this particular model for the DNA. And this particular model proposed by Francis Crick and James Watson and Francis Crick, this was also very much based on, another basis was what? Erwin Chargaff's rule. See, Erwin Chargaff, he provided a very strong support to this particular model, or to explain the particular model called double helical model. What was that support? He said that, see, there are 4 nitrogen bases that you already know, A, G, C and T, that is adenine, guanine, cytosine and thymine, which are bifurcated into purines and pyrimidines. See, by the discovery, by the experiments of Erwin Chargaff, it came to light that the A always pairs with T. That is, adenine always pairs with T with a double hydrogen bond. Nenipitkolli, this is a very important thing for you to remember. A always pairs with T with a double hydrogen bond. That was first given by whom? Erwin Chargaff. And another important discovery in this regard was what? G, that is guanine, always pairs with C with a triple hydrogen bond. Not to get confused between these two. G and C have a triple hydrogen bond, A and T have a double hydrogen bond here, right? See, this is what is called as complementarity. Okay, complementarity of base pairing. See, this is called complementarity of base pairing, right? And not only that, he also suggested, see, already I have written it here, he also suggested that the proportion of the purines is always equal to the pyrimidines. That means the proportion of A and T will be always equal, the proportion of G and C always be equal, right? And it is also true that, see, when A always pairs with T, A is what? It is a purine. T is what? It's a pyrimidine. A always comes with a, that means always, even in this case, G is a purine and C is a pyrimidine, right? That means always a purine pairs with a pyrimidine. This also means what? Purines are double ringed structures, pyrimidines are single ringed structure. ಒಂದು ಕಡೆ ಡಬಲ್ ರಿಂಗ್ ಬಂದಿದ್ರೆ ಮತ್ತೊಂದು ಕಡೆ ಸಿಂಗಲ್ ರಿಂಗ್ ಬರಲೇಬೇಕು. That means never two double ringed structures will pair up. Never two nitrogen bases with both double ringed structures will never pair up, nor two nitrogen bases with the single ringed structure also will never pair up, right? So that is how it is. So A always pairs with T and G always pairs with C. And also they are proportionate. In any DNA molecule, the purines, the content of the purines is always equal to the pyrimidines, and that is why he derives this particular equation, A plus T is equal to G plus C. That means, so it's always equal to 1. Right? So this is the background study for this double helical structure. So let us see what was there in the double helical structure. Okay, so let me draw the diagram for this double helical structure. See, after understanding the background of this one, how this was proposed and what was the basis for the proposal of this particular double helix model, let us now understand what is explained in this double helix model by Watson and Crick. See, let me explain with the help of the diagram. The first point, what does it say? The double helix structure of the DNA, that means the DNA molecule, it is made up of what? The polynucleotide chain, and in that polynucleotide chain, the backbone of the chain will be made up of sugar and phosphate. Okay, so sugar and phosphate will be in the backbone, and so nitrogen bases, they project inside. How is that? Let me try to draw the diagram. DNA diagram draw ಮಾಡ್ತಿರಬೇಕಾದ್ರೆ just don't draw it just like a snake. Okay, so there is a proper way of drawing here. Observe this carefully. Say I'm drawing here. It should be like this. Okay, so we need to make 2 strands of the DNA. Okay, so this is one strand, and while drawing the other strand, nodi one strand draw ಮಾಡಬೇಕಾದ್ರೆ I have drawn a S-like structure. You also should draw it like this only, okay? And while drawing the other strand, what you need to do is, see, you take it from here, and the other line should run parallelly. This should be a parallel line to this one, okay? And once again, when it comes here, this should be a parallel line to this one. See here, this is running parallel all along this parallel to this one, and once again parallel to this one. So why to draw it like this? See, once again like this. See, by drawing like this, what is achieved here, why the diagram should be drawn like this is, we will require the scientifically correct diagram here rather than beautification of the diagram. See, in this diagram, what are we seeing here? It should be, this groove is called as minor groove. This groove is called as major groove. Major groove and minor groove always should come opposite to each other. See, this is the minor groove, this is the major groove, right? This is the minor groove, this is the major groove, and always major groove and major grooves, they alternate with each other. Andre nodi illon major groove ide anta helidre immediately illilla, but illide, and another major groove comes here, right? So where minor groove is here, and the next minor groove is here, and the next one is here, and the next one is here. So alternating major grooves and minor grooves should be there, or in other way you can say major groove and minor groove, they should be opposite to each other. Okay, see, then pitkoli, this is the major groove. Okay, this is the minor groove. So in order to get correctly about major groove and minor groove, you need to draw this diagram in the same way which I showed you now. And see, the first point says is what? The sugars and phosphates, they make the backbone of the DNA model. So you know how we have already studied that in the previous session, how this will be having the sugars and they are connected with the phosphate group, sugar and phosphate, right? So like this complete entire model will be consisting of sugars and phosphates. Both the strands will be having the phosphate group. One phosphate group will be acting as a connecting link between 2 sugar molecules, because you know the third carbon atom of one nucleotide will be joining to the phosphate group, which is joined with the adjacent or the next nucleotide, and that is why it is going to form a sugar phosphate backbone here, right? So like this, and this also says us that the nitrogen bases, they project inside, because see, the nitrogen bases like this, see, maybe the cytosine, and cytosine should pair with which one? Guanine. So how many hydrogen bonds? Triple hydrogen bonds. Okay, let me take adenine here. Adenine always pairs with which one? Thymine, right? Thymine comes here. How many hydrogen bonds in between them? Double hydrogen bond. Similarly, so here if I take guanine here, it should come along with the C with a triple hydrogen bond. So like that, how many base pairs will be there? See, this is one base pair, this is one base pair, this is one base pair. So in one turn, this is one entire turn. So ಇದಕ್ಕೆ ನಾವೇನಂತ ಕರಿತೀವಿ ಅಂತ ಹೇಳಿದ್ರೆ, this is called one pitch. This is called one pitch of the DNA double helix. So in one pitch, approximately there are 10 base pairs. See, you are going to draw it like this. This is 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. 10 base pairs per each turn. Okay, see that I will come to that I will explain that in the fourth point. Okay, so ishtu artha aitu nimage. The backbone is made up of sugars and phosphates, and the nitrogen bases, they project inside to form the base pairs. Now the second point, they have got anti-parallel polarity. Anti-parallel polarity ant helidrenu, you know 5 prime and 3 prime end. So if one strand, if look at this particular strand, if this strand is running in 5 prime to 3 prime direction, idu 5 prime to 3 prime ide, okay, the other should be running in 3 prime to 5 prime direction. ಯಾಕೆ ಹಾಗೆ ಅಂತ ಹೇಳಿದ್ರೆ, let me draw a simple diagram here to explain you that. See, observe ಮಾಡಿ ಇದನ್ನ. See, I am going to draw. Just observe this simple diagram. You can also practice this in the same way. See, what are these arrow marks ಬರಿತಾ ಇದ್ದೇನೆ ಅಲ್ವಾ? But they are not actually the arrow marks. I am trying to draw the pentose sugars. Okay? Pentose sugar draw ಮಾಡಿದ್ದೇನೆ, and the first carbon atom of each pentose sugar is attached with which bond? N-glycosidic bond, and they have got the nitrogen base, either the double ringed or the single ringed or double ringed, like this. Okay, and the fifth carbon atom has got which one? The phosphate group, right? The fifth carbon atom has got the phosphate group like this. Yes, all of them have got, fifth carbon atom have got the phosphate group, and each phosphate group is attached to the third carbon atom of the next nucleotide like this, forming the phosphodiester bond, and this is one chain of polynucleotide, as you know, right? But ಇಲ್ಲಿ DNA ದಲ್ಲಿ there are 2 polynucleotide chains, and both of them are anti-parallel in polarity. Anti-parallel ಅಂತ ಹೇಳಿದ್ರೆ, one is running in 5 prime to 3 prime direction, the other is running parallel but in opposite direction. Okay, so how is that? See, I'm going to draw that also now. See, I'm similarly, just like how I drawn the first one, I'm drawing the second chain which is anti-parallel to the above chain. Okay, now the first carbon atom has got which one? The first carbon atom has got the nitrogen base. If here there is a double ringed structure, if this is a adenine, it should be thymine, or if it is a cytosine, it should be guanine, like that. Okay, so on one side there will be double ringed structure, on the other side there will be single ringed structure, like this. Okay, nitrogen bases are all attached, and now at the fifth carbon atom, once again the phosphate groups are attached. Phosphate groups at the fifth carbon atom, right? And they also are joined to the third carbon atom of the next nucleotide here. See, and what are we seeing here? Once again, the hydrogen bonds are being formed. See, if you consider this as A and this as T, there will be a double hydrogen bond, right? And if you consider this as G and this as C, there will be a triple hydrogen bond, right? A, T, double hydrogen bond, and G and C, triple hydrogen bond, right? A, T, double hydrogen bond. So, ಇಲ್ಲಿ ಏನು ನೋಡ್ತಿದ್ದೀರಿ ಅಂತ ಹೇಳಿದ್ರೆ, the third carbon atom here has got a free OH group, right? That is called a 3 prime end, and at the fifth carbon atom there will be a free phosphate group that is called as the 5 prime end. But look at on the other strand of the DNA where the fifth carbon atom is that is having the free phosphate group. This is called as 5 prime end, whereas the third carbon atom here is free. That is what is called as 3 prime end. So one is running in the 5 prime to 3 prime direction, right? And the other is running in the opposite direction. But both of them are parallel to each other. Yes or no? So that is why they are called as anti-parallel strands. One runs in 5' to 3' direction, the other runs in 3' to 5' direction. Coming to the next point, base pairs always are formed by hydrogen bonds. That means the nitrogen bases which are projected inside, they are forming the pairs with each other. That means purine always forms a pair with the pyrimidine, A always comes with a T and C always comes with a G, and they form what? Double bonds. In the case of A and T, triple hydrogen bond in the case of C and G. That point we have already noticed. The fourth point, see the DNA that we see here, the DNA that is explained by Watson and Crick is a right-handedly called DNA. ಇದಕ್ಕೆ ನಾವು BDNA ಅಂತ ಕೂಡ ಕರೀತೇವೆ. Okay, it's a right-handedly called DNA, and see the twist when you see it is coiled towards the right hand side. So it is a right-handedly coiled DNA. And in this right-handedly coiled DNA, so what is there? The pitch of the helix. This pitch, the length of the pitch of the helix is ಇದು approximately ಎಷ್ಟಿರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ 3.4 nanometer. Pitch ಅಂತ ಹೇಳಿದ್ರೆ ಹೇಗೆ ನೆನಪಿಟ್ಕೊಳ್ಬೇಕು? See, this is one X-like structure here. This is one more X-like structure here, right? So, or you can consider from this X to this X. This is one pitch. That means the length of one pitch is approximately how much? 3.4 nanometer. 1 nanometer ಅಂದ್ರೆ ಎಷ್ಟು? 1 nanometer is equal to 10 raised to minus 9 nanometer. Okay, 10 raised to minus 9 meter, sorry. 10 raised to minus 9 meter is equal to 1 nanometer. That will be the length of one pitch. And in each pitch, I have already told you there will be approximately 10 base pairs, right? 10 base pairs per turn. ಒಂದು ಟರ್ನ್, ದಿಸ್ ಇಸ್ ಒನ್ ಟರ್ನ್ ಅಂತ ಹೇಳಿದ್ರೆ ಒಂದು ಪಿಚ್ ಅಂದ್ರೆ ಒಂದು ಟರ್ನ್ ಆಗುತ್ತೆ. ಅಲ್ಲಿ 10 ಬೇಸ್ ಪೇರ್ಸ್ ಇದೆ. ಸೊ ದ ಡಿಸ್ಟೆನ್ಸ್ ಬಿಟ್ವೀನ್ ಈಚ್ ಬೇಸ್ ಪೇರ್ ಶುಡ್ ಬಿ ಹೌ ಮಚ್? That is 3.4 divided by 10 will be how much? 0.34 nanometer. Yes, so that is what is the measurement of the DNA double helix that was given by Watson and Crick. Right? Not only that, the fifth point, the plane of one base pair. See, the plane of one base pair stacks over the other. Andre enu? See, this is one base pair. This is one base pair. This is another base pair. This is another base pair. Another base pair. Another base pair. See, ಇದು ಕಾಯಿಲ್ ಆಗಿರೋದ್ರಿಂದ, ಹೀಗೆ ಟ್ವಿಸ್ಟ್ ಆಗಿರೋದ್ರಿಂದ ಬೇಸ್ ಪೇರ್ಸ್ ಏನಾಗಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ they form hydrogen bonds. Hydrogen bond formation ಆಗೋದ್ರಿಂದ there is a certain level of what stability to the DNA model, to the DNA molecule, hydrogen, because of the hydrogen bond. But so because of the twisting of this, the plane, this is one plane, this is another plane, this is another plane, the plane of each base pair will rest on the other one. See, idu ಒಂದು base pair anta helidre, this is one base pair, this is another base pair, so this is leaning on this one, this is resting on this one, so that this also will contribute to the more stability of this particular molecule of the DNA. And that is what is the fifth point is talking about. Right? So this is all about the DNA double helix, about the discovery of the DNA or deciphering the molecular structure of the DNA by Watson and Crick. So this is a very important question, 5 marks question. Okay, so idu 5 statements ide, salient features of the double helix structure of the DNA. It's a very important 5 marks question. So you need to learn directly from the textbooks. 5 points ide, very easy points are there, and you study that and you understand that, and you should know all the 5 points. Right? In the next session we can continue with the other aspects of the chapter.